San Diego 6 News in the morning.
Well, there's so much cutting-edge technology here in San Diego and medical breakthroughs taking place here all the time that every once in a while it's nice to be able to pass one on to you that seems to affect so many people. Joining us this morning from the Salk Institute, Jan Karlseder, who is a molecular and cell biologist, to talk about a breakthrough in chemotherapy for cancer. What types of cancers are we talking about here? Quite a broad range.
Yes, this is a fairly broad range of cancers. This is like ovarian cancer, breast cancer, prostate cancer. So we're talking about a therapy that has been used widely for a large number of diseases.
Now, if I understand this correctly, people have been undergoing chemotherapy with this particular group of drugs for some 25 years without really completely understanding how they work, just knowing that they do work, but not totally understanding the mechanism.
That is exactly correct, yeah. These are drugs, and you probably have heard of the names of them, like Taxol or Vinblastine or Velcade. These drugs have been used for pretty much for decades, and exactly as you said, they work, but we had no idea how. And that is really what I find so exciting about this discovery.
So the side effects are pretty drastic on some of these things as well. And by understanding a little bit better how they work, will that somehow increase their effectiveness and maybe decrease the side effects?
Well, that is the hope. This is what really is next on the table to test out. But yes, you're right. The side effects of these drugs are pretty much horrible, and patients are incredibly uncomfortable when they have to undergo these treatments. I mean, we're all aware of the nausea and the hair loss that's associated with that. Our hope is that now that we understand the very mechanism of how these drugs work, that we might be able to develop much more effective therapies, which of course might allow us to lower the doses and therefore lower the side effects.
So without getting too technical, in layman's terms, can you explain kind of what this discovery is in terms of how these drugs work?
Yes, I can try that.
Perfect. Okay. Well, if my eyes glaze over, take that as a clue that the audience's eyes are probably glazing over too.
All right. So these drugs target something that we call telomeres. And the telomeres are the ends of our chromosomes. So you know the chromosomes is where our genetic material is, and they are linear strands. They have caps at the end, these chromosomes. And this is what we call a telomere. And it's very easy to imagine that a good analogy is a shoelace. We have those plastic caps at the ends, and once those ends fray out, those plastic caps, the whole lace is useless.
Okay.
The same thing happens to a chromosome. The telomeres get shorter and shorter when we age or when cells divide. And at some point they are so short they cannot function anymore in protecting the chromosomes. And that is when the cell basically stops to grow or dies. Now, cancer cells do something to circumvent that. They make the telomeres long again, and that is really enough for the cell to become immortal. And immortality, of course, is one feature of cancer cells. Now, this shortening mechanism in healthy cells and the elongation and immortality mechanism in cancer cells makes the telomeres a very attractive target. What we found now is that the drugs like Taxol and so, they inhibit cell division, they inhibit cells at the stage when one cell becomes two cells and more cells, which is pretty much what kills you in cancer. The drugs that are used inhibit the cell division process, and that causes the telomeres to become dysfunctional, and that means they cannot protect the chromosomal ends anymore. That sends out the signal, basically a stress signal to the cell, and that kills the cell.
You're good.
Well,
because I think we all just got a great lesson in cellular biology here, and it makes sense.
Thank you.
Nice to visualize the shoelace analogy. Very good. So where are we at in terms of testing this right now? Mice testing right now, and how far away from human trials?
This is really what we are doing right now, yeah. So now that we have this hypothesis, so basically we think we know exactly how the mechanism works, the next step is to develop models that are usually mouse models for humanized cancer, where we can test the idea that we can use synergistic treatment with these drugs, these mitotic inhibitors like Taxol, and drugs that target the stress response in those cells, with the idea that synergistic treatment might be more effective.
And as we wrap up here, folks always want a timeline, and I know these things don't happen overnight, but what are we talking about?
It's inherently difficult to give a timeline for that. Such tests can take several years, and honestly, it's impossible to put a timeline on this, but what I can tell you is that We will not be able to solve the cancer problem without understanding the basic biology, and this is where institutes like the Salk come in, and if we make such discoveries, at least there's hope.
And it's happening right here in San Diego. Jan Carlseder, thank you so much for the lesson and for the
Thank
update.
you.
Great to see you.